understanding the biological composition of the environmental particulate matter is important for the study of its significant impacts on human health and disease spread.
These bioaerosol sampling methods and analyses may have a wide application in many fields of study, such as environmental monitoring and airborne pathogen detection.
To determine the total particulate number, first equip an airborne laser particle counter with a sensor to monitor the temperature and relative humidity. Then, collect the particle matter by the air sampling port on the top of the airborne laser particle counter, and use the test modules inside the sampler to measure the size of each class of particle simultaneously every 5 minutes. To collect particulate matter samples by cyclonic aerosol samplers, set a cyclonic aerosol sampler to collect a sample flow of 323 liters per minute for a 6-hour collection period, and use the automatic cleaning function to wash the inside of the sampler 3 times with sterile water. Then, place the sampler alone on a shelf or floor and start the collection. At the end of the collection period, store all of the samples at -20 degrees Celsius, protected from light, until their analysis. For particulate matter collection by filters, equip a high-volume air sampler with 20.32 × 25.4 square centimeter filters, and set the sample to a flow rate of 1,000 liters per minute for the appropriate collection period. At the end of the collection period, store all of the samples at -20 degrees Celsius, protected from light, until their analysis. For biological